Okay. Yes, so good evening, sir and madam who are here. And as you all know, this is a free webinar series, Science Dialogue series. Anyone can attend, from UG student to PhD faculty scientist. In this speech, we have today very dynamic and young resource person, Dr. Sheth. He's assistant professor at FRIGE. FRIGE Institute is at Ahmedabad. And this is very, you can, working on rare disease. This institute is known in Gujarat. And his PhD from Newcastle University, UK. And most important thing is, in this 10 year, in this decade, he has received 8 crore rupees as a research fund. And he has also published more than 40 papers and several news article also, popular, you can say, uh, popular article for awareness. And he is guiding several MSc student and PhD student. And he doesn't have only one topic which you are listening today. He has very diverse experience in different domains of biology, genomics, precision medicine, and different domains, rare disease. And his father is also a big scientist known in Gujarat. Now he is handling, as per my understanding, he is handling that institute since few years. Am I right, na? So welcome. Aapka mic. And
now you can. Now I
yes,
think you can
yes,
hear
yes,
me.
yes, yes. So thank you for accepting our invitation and welcome in this Science Dialogue series. This is
Thank you,
not
sir.
registered organization, but you know that more than 100 lectures we have conducted, and more than, I think, 3,000 participants have attended. Maybe some students, they are attending multiple times, but we have not restricted it from all over world. Uh, Attendees are from all over world. Anyone
Yes.
can attend. So with this brief, I welcome you and I request you to start your presentation because you have to go after 6:30. Anna?
Yes, sir.
That's why
So
30-minute preponed. We have preponed this lecture. Few participants may join. They keep on joining. Yes.
So thank you so much, sir, for the kind invitation to be here. And I absolutely understand that it's Saturday evening and you guys might want to do something better, but a big kudos to you for coming here and listening to a topic that is gaining a lot of prominence worldwide. So I think at least some of you would have heard about autism at some point in their time, be it in the family or in newspaper, or in fact, last year was Autism Genetics Month. So clearly it has captured the public's attention. Now, before I talk, just a little bit about what me and my group are doing. So I spent nearly a third of my life in the city. It's called Newcastle upon Tyne. And there's a beautiful river that's known as Tyne River. And I worked at this institute um, since my undergrad degree, finished my PhD and postdoc as well. And after spending 11 years, I came back to Ahmedabad. So I work at this institute. It's FRIGE Institute of Human Genetics, and it's been around for nearly 31 years at this point. And in my career, of which is, I think at this point, spanned nearly 12 years, I've worked with a lot of luminaries and pretty diverse areas. So worked on the pharmacogenetics of warfarin, which is one of the top 5 commonly prescribed drugs in the world. Worked on rapid molecular diagnostics using silicon nanowires with a company known as Quantum DX, which is now not a startup, but in fact, it has more than 100 people at this point. I've worked on infertility, male infertility, to be more precise, on cancer prevention, and coming back to India, I've worked on microsatellite instability testing, lysosomal storage disorder, biobank development, autism spectrum disorder, newborn screening, and repeated fetal loss. Something that we just started this year, so it's in its infancy right now, but we are developing a lot of interesting data at this point. So, without further ado, the whole point of genetics, in my view, is very neatly summarized by this gentleman, Thomas Adams, from 1618, whereby he said that he's a better physician that keeps diseases off us than he that cures them being on us. Prevention is so much better than healing because it saves the labor of being sick. And when it comes to genetics, it's absolutely true because it gives us the ability to predict whether somebody is going to have a particular disease. And accordingly, either we can keep things ready in terms of prevention of the phenotype from coming through, or keep the treatments ready. Now, this is just a snapshot of our nearly 20 years of experience of working in rare disease. So at this point, you're talking about more than 300 different rare diseases that we have worked on during our 31 years of existence. And this was a very neat summarization of all the different sets of diseases we have come through. There are more than 20,000 patients that are summarized in this paper. And in fact, this is the very first paper from India that talked about the prevalence of different rare diseases in different parts of the country. So a worthwhile read if you are interested in rare diseases. But coming back to the topic of, at hand, which is autism spectrum disorder. Now, inherently, it's a neurodevelopmental disorder that affects social interaction, communication, and lifestyle. And generally, the kids that come to our clinic present around the age of 2 to 3 years. I think the earliest we have seen is about 1.5 years. And the reason why it's called a spectrum, because the symptoms are highly heterogeneous. You actually see a variety of different symptoms coming through. And within that variety, you also have a degree of severity. It can be syndromic, it can be non-syndromic, and I'll explain about that in just a moment. But the scary statistic is that the worldwide prevalence is estimated to be about 1% of the population. So essentially, if you were to pick any 100 people in your community, you will at least find one person with autism on an average. And for some reason, it tends to affect males far more than females. You essentially see 3 males being affected for every 1 female child being affected with autism. And there are several theories or mathematical models that explains why it is so. But at this point, there's no genetic reason to explain why it is happening. In India, if we were to take prevalence estimates, it's about 1 in every 1,000 child around between the age of 0 to 15 years, which is likely to be having autism right now. So that's a pretty scary statistic if we were to think about it. It's somewhere in the ballpark of 2.9 million kids right now in this country with this disease. Now, generally, when they present to the clinic, they look something like this. So can you see the anxiety that this child is having? Generally, kids with autism tend to be extremely anxious. They can't manage social interactions very easily. But then you have something like this. This sort of movement, this is known as hand wringing. Sometimes they even have certain movements like this. This is known as stereotypy. So these kids have very peculiar symptoms. They would really look at people in the eye. They would barely speak, but they would have subtle symptoms, as I just presented. Now, when it comes to the genetics of ASD, it's not something new. People have been working on it since the 1980s. And most of the time, way back then, because karyotyping was the only technology available, people would look more from a chromosomal standpoint if there was something there. But as molecular technology started to come through, the very first disease that was identified was Fragile X, which is exceptionally common in male kids only because it's an X-linked disease. And as we went along, we started to identify more diseases such as UBE3A, which is your Prader-Willi-Angelman syndrome, that's on chromosome 15. You have MECP2, which is Rett syndrome, exceptionally common in females. Again, it's an X-linked disease that you don't strangely find in males, because in males it's lethal. And then a whole host of different genes have been identified since the introduction of sequencing, microarrays, and so on and so forth. So clearly, molecular genetics has driven the understanding of autism in the past 2 decades. And one thing that we have identified is that it's not just that you have a variant and you end up with a disease. The biology is a little bit more complicated than that, because there are instances whereby you would have the variant in just the child, but it's not inherited from either of the parents. They are de novo variants. They are highly penetrant. By that, I mean, if you have that variant, you're most likely going to have that autistic phenotype. So those sort of occurrences you do find quite commonly in autistic kids. But then there are scenarios whereby both parents are carriers of a medium risk or medium burden variants, and they are heterozygous carriers for it, so they don't have the disease. But when both alleles come into the child, you end up with the phenotype. And then you start moving towards a whole range of combinations, such as a medium risk variant with a high risk genetic background, or purely high risk genetic background, because of which you end up developing autism. But as you go from left to right, and you can see in that infograph, you see that more and more variance starts to come into play. So this is what makes autism exceptionally challenging to understand. It's not just one variant and one gene that leads to phenotype, but it's a collection of variants in a collection of genes that work together that leads to this phenotype. Now, way back in 2013, this is where actually things began, because the American College of Medical Genetics came out and said, anytime you see a child with autism, the first thing you do is karyotyping, that is, look at the chromosomes. Then you do Fragile X testing in males, because it's exceptionally common. And then you simply do microarray, and that's it. And the reason why that was the case was because microarray had just come into vogue around 2005, 2006. Exome and genome sequencing was still very, very expensive at that point. So microarray was recommended. And since then, this guideline has been stuck right there, right where it is. It has never gone through revision. But India is a little bit different. You don't have a heterogeneous population. In fact, you see 4,000 different endogamous populations that persist in this country. And because they are endogamous, they marry within the same community or within the same village. So the risk of having a recessive variant coming through is exceptionally higher compared to the Western population. The problem, however, is that till 2023, nobody had done a systematic evaluation of genetic architecture of autism. The ACMG guidelines were still in place. In fact, even today, they are in place despite all the evidence to the contrary. There is no genetic testing guideline based on Indian data. But well, the Indian data is now available. The guideline is currently being written up. So the hypothesis was that because India practices endogamy so excessively, you would see a higher burden of recessive variants in ASD patients. So way back in 2021, together with Dr. Sheth and Dr. Deepika Jain in Ahmedabad, we started to study the first systematic genetic architecture of autism in India. And for that, we recruited 101 patient-parent trios. So this is where you don't just recruit the patient who has the disease, but you also recruit his parents, his or her parents, to understand where did the variant actually emerge, or where did you inherit that variant. So across all of these 101 trios, we carried out karyotyping, and there were 73 males. So we also carried out Fragile X testing. If they were all normal, we did microarray analysis. So we wanted to see whether what ACMG guideline was saying was in fact true or not. And in those, only 3 CNVs were detected. So 3 patients were excluded from any further analysis. And in the remaining 98 patients, we did whole exome sequencing followed by segregation analysis. And here is a very broad stroke results that are being presented to you. you generally see a male to female ratio of 3:1, which is exactly what we were observing in our study. Our ratio was somewhere around 2.6:1. The average age of diagnosis was around 5 years. And probably since 2021, we have started to see that average age going down because people are suddenly realizing there might be something wrong with my child at a much earlier age. So the age of presentation is reducing quite rapidly. 61% of the couples were endogamous. So as predicted, a lot of them were practicing endogamy, and about 8% were practicing consanguinity. This is where you marry within the same family in itself. All the patients that we had on our study had developmental delay. It means the development of their walking skills, their talking skills, their thinking skills are not aligned with their age. Their mental capacity or their capacity to do any activity is far lower than what you would estimate at that given biological age. They had speech delay and they also had intellectual disability. So let's begin with the microarray analysis, because this is what ACMG guidelines say should be done. So out of 101 patients that we had, only 3 patients ended up with a CNV. And out of those 3 CNVs, 2 were deletions. One was present on chromosome 15, the other one was present on chromosome 9. And one was a duplication, which was present on chromosome number 1. All 3 variants were in fact pathogenic and they were de novo. That means the variant were only present in the child, they were not present in the parents, or they were not inherited from either of the parents. Ergo, the diagnostic yield of microarray technology is just 3%. Doesn't sound particularly high. But then we thought, wait, whole exome sequencing can be used to call CNVs as well. The only problem is, back in 2013, 2015, such mathematical algorithms didn't exist. But come 2021, things have changed. So we collaborated with Dr. Aneta Mikulasova, who is based at Newcastle University at the moment. And we developed a tool known as CNV Robot. Essentially, it allows you to convert the whole exome sequencing data into a microarray style data that you see right in front of your screen at the moment. And it tells you whether you have deletion or duplication. And at the same point, it also tells you whether you have got loss of heterozygosity. So it gives you double confirmation in case there is a deletion. So here is a very good example whereby if you see there is a sudden drop in the log2 ratio, it essentially say that there is a reduction in the number of reads in that given region, and correspondingly you are seeing only homozygous calls because you only have one allele from where all the SNPs are being called. So it gave us assurance that yes, it is doing what we assume it should be doing. Following that, we did whole exome sequencing on all 100 patients, and lo and behold, the 3 CNVs that were identified with a microarray, we are able to identify it using whole exome sequencing as well. But surprisingly, there was one case where in fact there was a deletion on chromosome 15, which was missed by microarray, but was picked up by whole exome sequencing. And when we did segregation analysis, it did turn out to be a de novo variant. The reason why this was missed was because this was a very small deletion, way below the detection range of a microarray, and that is the reason why it was missed. Nonetheless, it showed that exome sequencing can recapitulate everything that microarray is able to do. Now, following that, we wanted to see if single nucleotide variants, that means a single letter difference in the DNA, can explain why autism is so common. And indeed, in 27 patients, we identified 31 pathogenic or likely pathogenic variants, which were causative for autism. In fact, when we started to stratify by the mode of inheritance, we showed that majority of the variants were in fact missense variants. Majority of them were dominant, were present in dominant genes. And when it came to which genes they were hitting the most, it turns out that MECP2 was the most common gene. It means a huge chunk of these patients had Rett syndrome. And I'll tell you why this particular observation is so important. Along the way, we actually identified a brand new disease that had never existed before in the medical literature. And overall, if you were to include both SNV and CNV together, the yield of whole exome sequencing was 31%, which is, well, 10 times higher than what microarray could do. When it came to which biological pathways that these genes were involved in, it turned out that they were either present in synaptogenesis. So essentially, when neurons talk to each other, they talk via the synapses. These are the end of those neurons. But if the synapses are not formed properly, they are not able to talk properly to each other. So that was one crucial identification. The second one was chromatin remodeling. So this is where, again, Rett syndrome is one of the biggest chromatin remodeling genes, signal transduction and transcription regulation. So essentially, you see that a huge amount of multisystemic dysfunction is happening in autistic children. Now, as mentioned, MECP2 was quite peculiar because we identified one male child who had Rett syndrome. Now, as I mentioned before, Rett syndrome in males is lethal because you only have one X chromosome. You don't have another X chromosome to compensate for what's happening on the first chromosome. So the peculiarity was the reason why this child survived because it was a mosaic variant. It means that only some proportion of cells in his body had the mutation. The remaining cells were absolutely normal. When we checked the parents, of course, the parents were absolutely fine. So that confirmed that this mutation is what we call as post-zygotic de novo variant. It means that the variant was not there in the very first cell of the zygote, but once the cells started to divide within the zygote to form the fetus, somewhere along the way, you had this mutation emerging. And because of that, it got entrenched in a whole host of cells, and the child ended up having Rett syndrome. In fact, so rare it is that this is the only 12th case reported worldwide, and this is the first case of this kind from India. Now, in terms of one recessive new disorder that we identified, here is a case whereby two siblings had a very peculiar phenotype. It turns out that both of them had autism, but at the same time, they were congenitally deaf. They just couldn't hear anything. So the doctor did a whole host of MRIs, and what it turned out is that in your cochlea, there are nerves at the back, and it's the innervation process. It means when the nerves are forming around cochlea, that those nerve formations were not happening properly, and because of that, they couldn't hear. So when we did a lengthy analysis of these two siblings, it turned out that they had a homozygous mutation in a gene known as NEUROG1. And parents were carriers of this variant. Now, NEUROG1 is a transcription factor. Is a transcription factor, but because this was a nonsense variant that was identified in this case, we initially predicted that it would lead to nonsense-mediated decay. That means only a small chunk of protein is being formed, and the rest will be chucked out. It turns out, though, that in this case, nonsense-mediated decay doesn't happen, and you end up with something known as phenotypic pleiotropy. It means that you don't just get one phenotype, you get a whole host of different phenotypes. Not only that, NEUROG1 interacts with several known different autism autism genes. And because of that, you didn't just have a hearing loss, you also had autistic phenotype. And this was the fourth case from the globe, and this was the first case to be described with autism spectrum disorder phenotype. And in fact, on 11th of August 2023, this disease got listed on the OMIM database. So OMIM database is a collection of about 7,000 different diseases that are known to man as of today. And now this is one of those 7,000 disorders in the database. Now, as I said, you don't just think about single letter differences or copy number variation. There are quite a lot of different types of variants that occur in the DNA. They are called a structural variation. You can have big chunks of DNA that are missing. You can have dispersed duplication, tandem duplication, inversion, translocation, and so on and so forth. And all of these different sort of variants are exceptionally difficult to be detected by what we know as exome sequencing. The reasons are pretty complicated to be explained over here, but just bear in mind that short read sequencing or exome sequencing technology that we use today cannot recapitulate any of these different types of variants. So in 2023, we started working on understanding whether complex structural variants can in fact play a role in autism genetics. And for that, we started using Oxford Nanopore long read sequencing technology. So today, when you do next generation sequencing using Illumina, say whole exome or whole genome, you essentially chop down your DNA into very small bits, and you're sequencing only 150 letters of the DNA at a given time. When you're using long read sequencing, you are sequencing 10,000, 20,000 letters of a DNA at a time. So if there is, in fact, an inversion or a translocation that has happened, it's far more easier to detect that sort of event using a technology like Oxford Nanopore. So if you would remember this pathway, we had solved 31 cases. We had the remaining cases with us, and in those we picked 23, whereby we carried out long read sequencing. Now, generally, when you do long read sequencing, the result looks a lot like this. These are what are known as squiggles. And these squiggles are then computationally analyzed and base called into A, T, G, and C. You would then align your read against a reference genome. And using a combination of 5 different variant callers, we ended up calling all different types of structural variants. We annotated them, means we told how common or how frequent these variants are they aligning against a known ASD gene, and so on and so forth. And then finally, we carried out variant filtering and then prioritization. The critical thing is, generally, when you're doing whole genome sequencing, people tend to do sequencing at 30x, means each part of a DNA is read at least 30 times. But in our case, we went for 10x sequencing depth. The reason for that is that we already had carried out benchmarking that gave us an idea that if you were to just read your DNA 10 times or at 10x coverage, you would not have a significant loss in your ability to call structural variants. So on an average, when we started doing our sequencing across these 23 samples, the number of letters that we would read at a given time was about 6,700 letters. In a given sample, we would get around 176,000 different structural variant calls. And we identified only one case where a structural variant may cause autism. And here is that case. We have, this is a pedigree whereby these two siblings, they're twins, and we identified an inversion. It means a part of the DNA has just flipped around. And this inversion is occurring in a gene known as SNAP25AS1. Now, SNAP25 gene is intolerant to inversions like this, and it's also involved in synapses in the neuron. So clearly, this is a known autism-related gene, and it has previously been associated accordingly. The only problem is when we started doing segregation analysis, it turns out that this inversion is also present in the mother, but the mother is absolutely normal. So the question is this genuinely causing autism? Or is there some maternal protective effect that we are unaware of? Or is this variant simply contributing towards the phenotype, and there is some variant or there is some sort of mechanism which is also contributing towards generating this autism phenotype? It's very difficult to know at this point, but this is how far we have gone at this point. And this wonderful work was done by my PhD student, Janvi Shah. So in summary, microarray has a low diagnostic yield in Indian ASD cohort. In fact, it turns out the majority of the literature from around the world is showing similar diagnostic yields using microarray. When it comes to whole exome sequencing, we are seeing nearly 10 times higher diagnostic yield, primarily driven by single nucleotide variants. Nearly 70% of the variants are de novo in origin. It means these variants squarely occur in the patient. They are not inherited from either of the parents. And in fact, when we looked into all of these numbers, it turns out that regardless of which population or ethnicity you belong to, these proportions remain exactly the same. When it comes to structural variants, The role of structural variants in the etiology of ASD is likely to be evaluated, although there are different cohorts that are being analyzed throughout the world that we know of, but they haven't come out with their data yet. As far as the guidelines go, the ACMG guideline clearly at this point is outdated, and it is high time that with all the data that we have today, it goes through a revision. As far as the pathways go, majority of the genes are involved in synaptogenesis, signal transduction, transcription regulation, or chromatin remodeling. And all of this wonderful work, this is just a piece of work that we have done at the Institute. As I showed, we are working on several different diseases at the moment. But all the work has happened because of these wonderful advisors, team members, clinical collaborators, international collaborators, our collaborating organizations, and of course, the funder, the biggest funder being the Gujarat State Biotech Mission. And with that, thank you all.